let's see. Okay. We meet the first Tuesday of every month. Let's see. All right, there we go. And I have a speaker. Today's speaker is Chris Best, and Chris is the state botanist for U.S. Fish and Wildlife Services, and he's going to speak with us tonight about plant propagation. Before we get on with Chris's presentation, I would like to share with everybody what our scheduled events are for our San Antonio chapter. The third Saturday of every month, and we've been doing this now for several years, we have a work day on the Kirchoff Prairie where volunteers come out and we do various restoration projects. We meet, as I mentioned earlier, the first Tuesday of every month where we have a speaker on some subject that's interesting to us. Next month, September 7th, our speaker will be Brian Laughlin. He is the author of a book, Grasses of the Texas Hill Country. And we have a very special work day planned for Saturday, September the 11th at the Kirchoff Prairie. This is just a few miles east of Floresville. We're going to plant several hundred cells in trays of Manfredus seedlings. Our chapter is working with the National Butterfly Center with a goal to establish about 500 Manfreda plants on each of the Kirchoff Prairie and the Kirchoff Prairie. And we've collected a bunch of seed, and at our work day a couple of weeks ago, we planted about 600 cells at the Kirchoff Prairie, and we're really having some great results. We probably have close to 100% germination in those cells. So everybody that's on this Zoom call today, put down September 11th on your schedule, because this will be a great opportunity for us to implement some of what we're going to learn from Chris Best this evening on how to propagate native plants. Another very important event you might want to put on your calendar is Saturday, October 7th. We are hosting a bird skills workshop. It's being conducted by the Bear Audubon Society. Patsy and Tom Inglet are leading that workshop. We'll spend 2 or 3 hours in class session out there on the prairie, and then the balance of the day observing bird populations on the prairie. So with that, let's move more into what Chris Best is going to talk about. I'd like to say that we are really proud to have Chris speaking with us tonight. Just this week, he is being awarded the 2021 U.S. Fish and Wildlife Services Regional Recovery Championship Award. This is a very significant award in U.S. Fish and Wildlife Services, and Chris is being awarded that here in our region. And his presentation will be on, as I mentioned earlier, native plant propagation. Rather than talk a bunch about Chris's education and the many things he's accomplished and articles he's written, I just would like to briefly say what I have experienced with Chris. More than a dozen years ago, when we began trying to restore our farm to a prairie, we met with great failure, and we were asked to contact Chris and get some advice. And I still remember that first meeting with Chris, where we described what we were doing and the outcome we were getting. And I still remember Chris looking at me, and in his slow Texas drawl, he said, "You know, Don, these native plants did quite well for thousands of generations without the benefit of modern farming techniques. Their seeds simply fell on the ground, and they may have been stomped into the earth by animal hoofs, giving them tight soil seed contact. They like that environment, and they've done quite well." Well, we know that with 1 or 2 centuries of human intervention, we don't necessarily have that environment anymore, but we can get close to it. And that's what Chris has been helping us on our property do for about a dozen years. I would say Chris is a walking encyclopedia, so we have a great deal to learn from him this evening. And he suggested that this presentation be interactive. So during his talk, if you have a question, first of all, let's keep our audio and video turned off. But if you have a question, turn it on, ask the question, and Chris would like interactive discussion during his presentation. If you'd rather write your question into the chat, the chat box, do that, and we'll interrupt Chris. So with that, Chris, why don't I see if I can turn my computer off and yours on, and we'll turn the meeting over to you.
Okay. Let's see, hang on a second. Host has disabled participant screen.
Oh, you know, I was supposed to click something.
Yeah, go up to my picture and you see where the three dots are.
Yes.
And
let's see. They're making you the host. Sorry about that.
No, not at all. Let's see now. I'm going to choose multiple participants can share simultaneously since I'm the host. And let me try this. Share. Can you all see my PowerPoint?
It's coming on. There it is.
Okay, good. I'm going to go to slideshow from the beginning. There you go. You should see my opening slide there.
Yes.
A black screen with a butterfly. And I will reiterate, well, first, thank Don for his very kind exaggerations. And, you know, it's been part of my job has been to work with private landowners and other organizations and help people achieve their goals. And I was telling Don earlier today, I think that's been the very best assignments that I've had with the Fish and Wildlife Service. And I also want to reiterate what Don was saying. And actually, you know, now that we've been in this pandemic a year and a half, and a lot of us have, you know, I used to give presentations to the public fairly often. And now we've gone to Zoom or Microsoft Teams. And it's a little different. And because I can't see you all, and especially if you're giving like an hour-long presentation, doubts start to creep into your mind. You know, is anybody really listening anymore? Are they interested in this? Do they get my jokes? And so I think it's, well, we've all sort of discovered it's very helpful that people raise questions as they have them. And Don is going to be our clearinghouse for any questions that come in via the chat. And he has instructions to stop me at any point and introduce questions, or as he mentions, you should be able to turn your audio and video on and just speak. So anyway, today's presentation is part of a series that I've put together on related topics ranging from monarch and pollinator conservation, milkweeds, pollinator gardens. I have a presentation on pollination itself, habitat restoration, native grasses and grassland ecology, and this one on native plant propagation. And all of these presentations, now that we've made them, you know, you all helped paid for them. And so these are public domain. And if you would like to use any of these presentations or the photographs, just let me know and we will send them your way. You can use them as you wish. So the purposes and benefits of native plant propagation include habitat restoration, wildlife habitat restoration, the conservation and recovery of rare plant species. Some of the benefits include the fact that when we use native ecotypes of native plants, they are adapted to the soils and the climate of a specific area. Plant propagation also is a very useful way for us to learn more about rare species, including a lot of things we can't learn from the plants in the wild without destroying them. But plants do have this advantage that typically they make seeds, sometimes lots of seeds. We can propagate them and study them without having an impact or a further impact on the wild populations. Using native plants gives us an alternative to filling landscapes up with introduced species, some of which become invasive. And one of my favorites is I get so bored of the big box plants, big box store plants that everybody uses in their landscapes to utter monotony. I mean, if you love plants, we have so many wonderful, useful, beautiful native plants here in Texas. Why can't we use those in our landscapes and have something a little different? Now, on the downside, native plants can be a lot more difficult to germinate and to propagate than native species. There are rarely going to be commercial sources for native plants, particularly local ecotypes. We want to make sure we don't over collect seeds of native plants, because we could be, especially if they're rare species, because we could be making them even more rare. Another concern is if we collect a very small sample of seeds of a rare plant species, and then we start propagating these and then multiplying them and selling them and planting them everywhere, then you over amplify a very small segment of the species' genetic diversity and basically swamp the wild gene pool. And that can have very negative consequences, especially for species that are obligate outcrossers. And once you have too much of one genetic type, the plant may no longer be able to make seeds. So that's a very bad situation. And also, we have to be very mindful that repeated cycles of propagation is a form of genetic drift. And it's very easy. It only takes a few generations to start turning a wild plant into a cultivar. And one of the first things that's lost are dormancy mechanisms that wild plants have evolved that enable them to survive in a place like Texas, where the climate is, and especially rainfall, is extremely variable. My first experiences growing native plants was as a Peace Corps volunteer in Guatemala in the 1980s. And I was sent to this place called Comapa, which was a pretty isolated municipio right on the border with El Salvador. And my job was to start an agroforestry program there. And the people I met there in the villages had never seen a gringo before. And initially, a lot of people were kind of afraid of me. I found out later they thought I could give them the evil eye. And but eventually, when they found out I didn't spread evil eye, it was almost hard to get any work done, because everywhere I went, people wanted me to work in their village. Most of the people had little or no education. The gentleman on the left, Lorenzo Ramirez, I selected him as our permanent. So I was a temporary person there to train people. He became the permanent agroforestry promoter, and was one of the most remarkable people I've ever met. He taught himself how to read when he was 39, and was a natural leader. And anyway, and I learned so much from him and all of these people. But one of the things that really sticks with me is, although these people had little or no education, they were smart as hell. I mean, they were very intelligent and keen observers of everything around them, and wonderful people to work with. But anyway, so you know, we wanted to produce the species of trees and other native plants that they found valuable, but that had become depleted on the landscape. And that's something poverty will do. It will deplete landscapes pretty quickly when people are desperate. And, you know, to give you an idea of the extent of poverty there, Lorenzo told me that if 2 out of every 3 children survive to adulthood, they thought they were doing pretty darn good. So, you know, 33% infant mortality rate is something to think about. So we grew species like Matiliscuate and Guachipilin and Conacaste, and you can see the tree on the right is Aguaje, or Leucaena leucocephala, which in some parts of the world has now become invasive, but this was one of the really good native trees in their area. It's very fast growing. That tree there was 2 years old, and a terrific species for firewood, which these people were in constant need of. It also was a nitrogen fixer that added nitrogen to the soil. The leaves could be used as forage for cattle, a really useful tree. So everything that we did, we had to learn how to do. Nobody knew how to do this. There weren't any manuals. And we just figured stuff out on our own and used whatever materials were available. And that's what we call appropriate technology. After I came back to the United States, beginning in 1990, I was very fortunate and I got the job of a plant ecologist at Rio Grande Valley National Wildlife Refuge, which is in the 4 southernmost counties of Texas, right on the border with Mexico. And it was an interesting segue because what they wanted to do was take retired farmland and restore native vegetation, forest, thorn forest, shrubland, savanna, to create wildlife corridors in an area where only about 5% of the native habitat remains. And the idea of these corridors was to connect together all the little isolated fragments of habitat that were left here and there. So in this photograph, an aerial photograph, you can see right at the edge of Brownsville, we had a tract of land called Phillips Banco. And you can see the Rio Grande and Mexico at the bottom. And so all of the fields you see there were crop fields. Some of them had already begun being restored when that photograph was taken in 1994, I believe, or 95. And 15 years later, it looked like this. And you can see that it was well on the way to becoming a subtropical forest and was serving as habitat for chachalacas and green jays and hook-billed kites and all kinds of interesting wildlife. And we hoped that ocelots and jaguarundis would be able to use these areas. Now you can see each one of those colored polygons represents a different field that we planted at a different time. And they were all, you know, I have data on every single one. We had a palette of over 60 native species that we used every year. And we could select the trees and shrubs that were most appropriate for each site. All the seeds came from the Rio Grande Valley. They were all locally adapted ecotypes. And so we knew they would do well where they were planted. If you look at the the field in the very center, and you see some black lines on it. I'm going to zoom in on that. And I used that field to test or do a pilot reintroduction of two of the rarest species in the Rio Grande Valley. One is a listed endangered plant, Ienia limiparis, also known as Tamaulipan kidney petal. And the other is absolutely the rarest tree in Texas, Esenbeckia runyanii, which we call limoncillo. There's only 15 of these remaining in the wild in Texas. And so we collected seeds and we introduced them. We started with 200, correction. In 2008, I went back, and so all the colored circles and crosses indicate where I found these species 10 years after we planted them. And the population of the Tamaulipan kidney petal had more than tripled. And the Esenbeckia trees, I had, an acceptable survival rate, and they were growing. You can see the limoncillo is on the right, and the Tamaulipan kidney petal is on the left. And the limoncillo has a very long lifespan, and it had not yet begun to flower or reproduce, but the Tamaulipan kidney petal was flowering and cranking out seeds. And as I say, there was 3 times as many individuals as I had planted 10 years earlier. So that's the objective. We wanted to create self-sustaining populations and habitats. In 2006, I was very fortunate to take my current position as state botanist, which is a bit of a euphemism because I'm really the only botanist our agency employs in the state of Texas. But it sounds impressive, and it sounds like I oversee some vast infrastructure, but it's just me. Anyway, continuing on the theme of using plant propagation as a conservation tool for rare plant species, in the photograph on the left, you can see pallid coneflower. I discovered this in a vacant lot across the street from our office in Austin. As far as I know, this is the southernmost location for this beautiful coneflower that has a range that extends all the way to Canada. And it wasn't even known this far south. And then here it was in a vacant lot. And I shouldn't admit this, but the place wasn't fenced or anything. You could just walk across it. People did. And I helped myself to those seeds. Now that it's breaking all the rules because we're supposed to get landowner permission always. But anyway, I saw the opportunity and I did it, and I have those seeds, and I'm looking for an opportunity to use them because this represents the extreme southernmost location for a species that has a range that may decline as climates change, and as climates warm, the range we expect may shrink northward. And so if you have a sample of its southernmost population, that may be a very important component of the species' overall genetic diversity. So I have those seeds, and a few months after I collected them, the landowner bulldozed the site, and that is now a 20-story office building and parking garage. So timely, right? And on the right, you see an event that took place last fall. Before the pandemic hit, there was a huge amount of construction of oil and gas pipelines from the Permian Basin to refineries along the coast. These pipelines are over 500 miles long, and they stretch, you know, state law or changes in state law allowed the pipeline developers to, they basically have eminent domain and they can put those pipelines where they want to. And some of those pipelines came through populations of a listed threatened plant species called the towbush fishhook cactus. But the developers did have to consult with U.S. Fish and Wildlife Service, and they have to come up with a plan that will prevent an incremental decline in each listed species due to their project. So in this case, these towbush fishhook cactuses were salvaged from the site and they were cared for at San Antonio Botanical Garden. And then in October, we planted them out in the field. And the consultants have reported back to me and sent lots of pictures. So far, every single plant has survived and several of them flowered this spring. Plant propagation begins with a collection of some sort of propagule. And, you know, typically we like to propagate plants with seeds because there you're capturing a lot more genetic diversity than if you're just using cuttings. Now, horticulturalists, they want to use cuttings a lot of the time because they don't want genetic diversity. They want every plant to look the same, have the same flower colors and the same growth patterns, because maybe that's what some landscaper has in mind. But usually when we're working with native plants, we want the opposite approach. We want to capture all the genetic diversity we can because our goal is we want self-sustaining, genetically healthy populations. So typically we're growing plants from seeds and also collecting seeds from plants in most cases really doesn't incur any harm to the plants that are there. Now, there's some guidelines, and I list some ideas there. Of course, you want to, if you're looking for native plants, scout frequently in as many habitats as you can access. Of course, always get landowner permission before you go out. Use your GPS to record where you've collected plants. And skipping down to the last button there, always label your collections with the species, the location, and the date. That's very useful information. We don't want to over-collect plants from a wild population because you could be contributing to the decline of a rare species. So the Center for Plant Conservation recommends that for very rare plants, limit collections to less than 10% of what a population would produce in a year. In my personal experience, it's a rare time when we can even collect 10% of the seeds that a population produces. You know, you can't reach all the plants. When you arrive at the site, a lot of the seeds have already shattered and others are not mature yet, and so on. So it's important to collect only fully mature seeds. Many plants simply won't germinate if the seeds aren't fully mature. Grasses are a really good example of that. There's no point in collecting grass seeds that are still green. And so the problem is when you collect seeds that are fully mature, a lot of them have already dispersed. But that's better than collecting seeds that won't germinate. We want, especially with rare plants, we want to capture as much genetic diversity as possible. So you may be tempted, you find this one individual that's just loaded with seeds and all the others have just a few. So you think, oh man, this plant is really healthy. I'm going to get all my seeds from that one individual. But the end result, would you be creating a population with very little genetic diversity, may not even be able to produce seeds. So try to collect evenly from as many different individuals as possible. And in the case of very rare plants, we actually keep the seeds from each individual separate. And then what we'll do is set up a seed production plot where every individual is labeled. So we know, okay, these 10 plants here are the offspring of plant number 21, and these are the offspring of plant 22. And in that way, we can make sure we're carrying that diversity forward into the seed production that would be used for reintroduction, reestablishing a wild population somewhere else. If we don't do that, the danger is that you think, hey, I collected seeds from 100 individuals, but most of the seeds came from 5 individuals. And then chances are, it may end up that you're sending seeds out to a reintroduction site that mostly came from 3 individuals. And then you find that that reintroduced population, you have surviving individuals, but they don't have enough genetic diversity to make seeds, and they don't become self-sustaining.
Chris?
Your picture, you got questions?
Yeah, we have a question here. What is the acceptable survival rate of seeds that you plant? Or what would be your target survival rate?
Well, you mean once they go into the field?
I'd say once they're in the ground and growing, what would be the survival rate?
Well, I'm not going to say that there's one answer to that, because different sites can be a lot more difficult to restore, and some species are notoriously difficult to restore or to get to survival. In our work in the Rio Grande Valley, which is a pretty harsh location, a lot of the sites were dry land. We had huge problems with invasive plants. And in a dry land site, you're dependent on rainfall. So we would try to get everything prepared, get the site prepared, control the invasives, get all our plant material, which we were mainly using seedlings, get everything ready, and you wait for it to rain. And sometimes it just, these thunderstorms come by, but they miss that site. And you're like, gee, my seedlings won't last forever either. What do I do? And sometimes you'd finally get one halfway decent rain, you'd go out and plant, and it just wouldn't rain after that.
Chris, would you mind checking the waiting room? There are people trying to get in, and I've lost control of that. Sorry.
Okay, I see a chat. Okay, participants, would it be there?
I think so.
I'm not seeing a waiting room. I see 30 participants. So, and if I look and I see an invite thing, is that anything? I don't know. That's to send invitations.
No, Julia, where does Chris need to look for the waiting room?
It should be under, it says participants, it says 30. The people who haven't been admitted into the meeting yet should be at the very, very top.
I'm not seeing anything. Oh, waiting room. I had to click on it. Admit all.
There you go. Sweet.
That'll do it.
Okay. I hope that, yes. Okay. Many apologies to those who were left in the waiting room. You didn't miss much. Yeah, there was a very interesting question. What's an acceptable survival rate? Well, in some places, if we got 60% or even 50% survival of our seedlings, we considered that acceptable under the circumstances. Oh, okay, admit. Under good conditions, we could often see 1 year survival of 85 or even 90%, and we thought that was good. Now, there's other environments where if all you got was 85%, you might not be that pleased, but these were pretty harsh conditions, and there are worse places in the world. But in general, the more arid a location, the more difficult it will be to restore. What else? I was once asked that question, by the way, by some of my colleagues with the Facultad de Ciencias Forestales, the Forestry Sciences faculty at the University of Nuevo León. And these people are experts and leaders in reforestation in Mexico. And I said, well, now I have some sites where I only had 50 or 60% survival after a year. But after 10 years, my survival rate was up to 500%. And they're like, what? How do you figure? And I said, because by that time, the trees were already reproducing on their own, and they made many, many copies of themselves. And they're like, what a great idea. What a great concept. So let's see, I see some chats. We got, okay, there was Manfreda. I already answered that one. Greetings from another PVC, Guatemala. We definitely have to communicate. I can't even see. Is that Sean Davis, or that's the next one? Or from Native Print? Okay, just please get in touch with me later. Sean Davis writes, what's your acceptable survival? Okay, we did that. And if you have missed some of the meeting, okay, we got that. All right. I think we're caught up, are we? If there's any others missing, let me know.
Yeah,
Okay.
we're caught up. Yeah, go ahead and proceed. I'll keep watching that.
Okay. Okay, let me get this out. Seed collection. So anyway, there's plenty of appropriate technology methods to be used for collecting seeds, which I'll talk about. Oops, no. There we go. Now, ecotypes are very important. So you may be interested in a particular species that's native to your area, but that species might range all the way from Texas to Canada. Ecotypes refer to the local populations of a species that are specifically adapted to the climate and soils of a particular area. And it's always best to collect seeds from an ecotype where it will eventually be planted. And in most cases, a native ecotype will do best there compared to ecotypes from other regions. And as a guide, we recommend, as a minimum, using the Gould ecoregions of Texas, which you can download from Texas Parks and Wildlife Department. Now, there are some cases where introduced ecotypes have become invasive. One example is the common reed, Phragmites, which is native to North America. But what happened is a European ecotype was introduced into the Northeast United States and has spread very aggressively. And I'm not sure if we know the reason why. And another possible example is tanglehead, a native grass of South Texas that is found in many parts of the tropical regions of the world. And beginning in the late 90s, it started to spread very rapidly in South Texas. And we don't know why, but it may be that a non-native ecotype came that somehow evades local pathogens. We don't know. Seed collection methods, be creative. You can see, like I mentioned, the problem of seeds shattering and dispersing on their own. With some of our milkweeds, if they're not ripe today, they'll be ripe tomorrow, and the next day they're gone. And so one way to make sure you can collect those is to use this. Some people use bridal veil, or I go to a fabric store and I'll get this sheer nylon drapery material. And I think it costs like, $7 per square yard or something like that. It's not that expensive. And you have to make sure you get the kind where the threads won't separate. And those make great seed drying bags, and also you can wrap them as you see around a maturing capsule. And you might come back months later and you can find your bag still wired to the stem and you've got your seeds. Another way to get seeds is to let animals do it for you. And then they get scarified all at the same time. Now, the technicians I formerly supervised were never very happy with the assignment. I don't know why. Usually I ended up having to do it myself. And one warning is definitely do wear a mask when you work with animal manure, because there are some zoonoses that we can get, we can get certain illnesses and parasites from animal droppings, so be careful. I'm going to move on. Now let's see what happened. Okay, I assume, oh, admit. I see an admit button here. I think I just admitted someone else. Okay. There we go. Seed processing and storage. Every species is different. For species like the Texas tuberose and the hill country penstemon that you see here, these seeds are produced in capsules that dry as they mature. It's pretty easy to separate the seeds from the fruit, the capsule. And you can use a variety of means like various sized screens, which you can make from things like a window screen, and you can go to the hardware store and get 1/8 inch and 1/4 inch and 1/2 inch hardware cloth, and you can wire these to a wooden frame. And in some cases, you can use those to either the seed passes through and you collect it below, or the seed is retained and some of the chaff will fall through. And then you can put a fan underneath the thing. And now you've created an air screen. You could buy an air screen from a specialist company for several $1,000s, or you can make one out of window screen and a fan, and you can even get a rheostat and wire it into that fan so you can fine tune the speed of the fan, and you can get it where you can just nudge that speed up and just to the point where the chaff is starting to lift off and leave the seeds behind. And of course, winnowing is an age-old technique. You can just let the wind blow the chaff away from seeds as you drop it from one bucket into another. Or you can set a fan up, which is blowing sideways, and that might be a better method because the wind speed will be more consistent. Believe it or not, in the program I formerly worked for, we actually did plant mesquite trees on purpose. And in order to get enough seeds, you can collect lots of mesquite pods, but how do you get the seed out of it? Well, I went over to Reynosa, Tamaulipas, and went to a hardware store, and for $15, I bought a hand-powered corn grinder. And by inserting washers into the mechanism that clamped the grinding plates together, I could adjust the space so it was just wide enough that I could grind up that pod and the seed would come through without being damaged. I don't know how many of you love chili piquins like I do, but even if you don't personally, chili piquins where they grow are a very important source of food for birds, and mockingbirds love them, of course. And you probably don't want to mash chili piquin fruits up by hand. Now, I should mention that the chili piquin, which is a native wild chili pepper, is one of those species that if you collect the seeds or the fruits when they're still green, those seeds won't germinate. So you definitely want to collect fully ripe red berries. And I like to just put them in a Ziploc baggie. And you can age them a few days to make sure they're good and soft, and then add some water to the bag, and then just manually you can rub it like that and you won't get capsaicin all over your hands. And so that if you happen to touch your eyes hours later, you won't be screaming in pain. And so you mash them up. And then another technique that works very well with chili piquin, once they're all mashed up and the seeds are loose, sort of pour all that into a mason jar and fill it with water, and be sure to let the bubbles finish rising. And then you can stir it a little bit and just gently pour off everything that floats. Or you may have to spoon it out initially, but then just pour all that off. And just repeat this process, maybe 5 or 10 times, and you will end up with pure, viable seeds at the bottom of the jar. And at that point, you can pour that out over a screen and dry it quickly. We need to store, if we're going to store our seeds for any length of time, you need to dry them thoroughly. And that means well beyond when they look dry. I've seen many a batch of seeds that looked dry, and people put them in a container of some kind and set them in the refrigerator, and you come back a few months later and they're all moldy. So as a general guideline, most native plant seeds you can air dry under ambient, meaning outdoor temperatures, provided that they don't get rained on. So it could be inside a garage or something like that. And usually at least a month. If you're able to, if you have enough seeds that you can destructively sample them, typically you see guidelines of 5 to 15% moisture content is necessary for long-term storage. Most people will store seeds in a paper container or a cloth bag at room temperature, and that's, for many species, that's good for 1 to several years. And the viability can always be extended when kept at cooler temperatures like a refrigerator. But just be very careful if you have seeds in a refrigerator, when you open that, if you take the container out while it's still cold and open it, you're going to get condensation on the seeds. So the next time you use those seeds, they may be moldy. So let the container warm up before you open it. And let's see, John, I'm going to take it on your advisement how long we can go. But so there are some, I'm going to compare and contrast in the following slides between sort of microscale propagation, such as for a landscape, or possibly if you had a nursery and you were selling native plants at some smaller scale, versus macro scale propagation, such as restoring habitats in large areas. And I see a chat question.
Yeah, Chris, before you get away from storage, sometimes pests can get in the seeds like weevils. How do you keep pests out?
Well, remember that some seed weevils actually are present, but they already got in the seed when you collected it. They were there before. But there are other seed weevils. So refrigeration is one way that usually stops most seed weevils. Some people, you may want to use something like, an insecticide. I prefer not to. And we generally found it wasn't necessary to use insecticides like, I think 7 is one that people typically use. But, if you're going to use an insecticide on seeds, definitely label the containers to warn anybody else that might use those seeds. Hey, you need to wear gloves when you handle these seeds and don't breathe the dust. There's a question about mesquite propagation. The loss of old mesquites in San Antonio neighborhoods, and there's no source. Well, you could just bring in a herd of cattle, because nothing spreads honey mesquite better than livestock. But let's see, in the interest of time, I'm going to forge ahead and I can discuss propagation of mesquite by seed. So Joan Miller can email me or anyone else. And my contact is at the end and beginning of this presentation. Let's see. So I'm comparing and contrasting methods used for large-scale restoration, where typically we are buying seeds in bulk quantities usually from a commercial seed company. You're going to seed at rates that typically range from 20 to 60 pure live seeds per square foot. So that's 1 3/4 live seeds per acre. Pure live seed is a standard used in the seed industry, and it refers to the quantity of basically of germinable seeds that are in the whatever you buy from the grower, and it takes into account the amount of viability and the amount of trash. And this is very important, especially with native grasses, because it's often not possible or practical to separate the actual seed from the structures that it was produced in, that the plant uses to disperse it. When planting direct seeding to large areas, the seed germination is dependent on environmental conditions. So you want to time that operation so the seeds are just getting into the ground at the beginning of the best period of time for germination. So in most parts of Texas, planting seasons can be like late summer, early fall, or in the spring. And as a general rule, often wildflowers may do relatively better in the fall and native grasses in the spring. But your site preparation are extremely important. You're not going to be able to find nearly as much genetic diversity in bulk quantities of commercially produced seeds, but it is a very economical method per individual and per unit of area. Containerized seedlings, typically we're growing plants in containers when we have very limited source of seeds, especially with rare plants, when very specialized methods are required for germination, when we need to get the greatest number of individuals per quantity of seeds. It's going to be much more expensive per individual, but you do have much more control over species composition and the spacing of plants in your restored area. How do we get a higher plant diversity when we're doing large-scale restoration? Well, we can combine both methods, okay? You can restore most of your area using direct seeding. And let's see, now for some reason, it's not, oh, there we go. So an example of smaller scale propagation, here you can see a native plant landscape that I put in. And the area you see on the left was, people had been parking their cars there and the soil was horribly compacted, and even Bermuda grass was struggling to grow there. So I dug it all up by hand and I brought in crushed limestone and I planted some of my favorite Edwards Plateau wildflowers, including some fairly rare plants like emery milkweed and the Texas tuberose that you all are familiar with. And oh, and I forgot I was going to offer you guys a quiz plant. And it was the very first, okay, well, if we have time, we'll get around to that, but I'm going to offer a prize for anybody who sticks out all the way to the end, because I've got a quiz plant. And it's in my landscape. Okay, here's another landscape we made at Santa Ana National Wildlife Refuge. the area had been a Bermuda grass lawn. It was boring. And it was the first thing every visitor saw when they came to the wildlife refuge. And we thought, man, that doesn't say anything to visitors about what we do. And so we herbicided out the grass and we tore it all up and we planted all these native plants. And it was really beautiful. And we had different themes. We had like a trail going through a buffalo grass lawn. And there were these islands that represented different themes, which were different habitat types. So as soon as a person got to the refuge, they could get out and, you know, maybe they're old and, you know, the trail we built was approved for disabled people. So maybe you can't walk 10 miles and go see all this stuff, but you could roll along on this approved trail and you could see samples of everything. And the president of the North American Butterfly Association told me about 2 years after we planted this, that in this one butterfly garden, he had seen more species of butterflies than in any other landscape he had ever seen. Getting back to the theme of the large scale restoration, you can see some of the equipment that can be used. In the photograph on the right, you can see a gentleman, Dr. Ashley McCallan, who asked me to work with him, and we planted 85 acres of native grassland on some old sorghum fields, or that he had come to the conclusion that in most years, the amount of hay he produced there was about equal to or barely, or some years it didn't even reach the cost of planting it. And so he got interested in using native grasses, which are more expensive to establish initially, but once they're there, all you have to do is manage them, and you can graze them or you can cut hay. And so we did this. Now, in that photograph, you see a piece of PVC lashed to the front of the tractor. That's another example of appropriate technology. It turned out his tractor didn't have a hydraulic system, which meant we couldn't lift the planter. So we had to plant the whole field concentrically, and it became difficult to line the tractor up. You were always having to look behind you to see if the planter was matching the last planted rows, but by then you would steer off course. And so we used that piece of PVC to guide us, and it worked beautifully well. As John will tell you, you definitely need to use specialized equipment for drilling native grass seeds. Don't use a grain drill. They will plant the seeds too deep, and essentially none of them will ever reach the surface. Here you can see another very small native grass drill that was built by our friends at Natural Resources Conservation Service. And John Riley, who was the director of the Plant Materials Center, not only loaned us the equipment, but he came out to plant, to operate it for us. And in the aerial photograph, what you see is this was an old pasture on Dr. McKellen's property that had been overgrazed for so long that there was almost nothing worthwhile left on it. And there was, since he had taken animals off of it, it was coming back, but he wanted to speed that up. And in particular, he wanted to improved habitat quality for pollinators. And so we got a mix of good pollinator species, and we planted, and we had to work around all the features of that site, which included, you can see existing live oak colonies, and there were a lot of rocks, and there was an arroyo and all of that. And that little 4-row planter was just the right tool for the job. Don, how are we doing on time? Can we keep forging ahead?
Yeah, I would say so. Now we've spent an hour, and I know that's the length of our meetings. So I guess anybody who is ready can drop out and just keep on until you finish, I guess, Chris.
Okay. A couple chats. Yeah, you're welcome to stay. And remember that prize, I'm not offering it until, I'm only offering it to people who last all the way to the end and who are still awake. Let's see, do you recommend removing seeds from all fruit and then drying, or can you dry small fruits? I've always found it better to depulp fruits and dry the seeds, because quite often when you dry fruits, some of the seeds may get moldy. It makes it a lot more difficult to store the fruits. Can you share the recording? Okay, I think Don will probably talk to you that about how you get the recording at the end. What type of containers to store seeds? Again, you know, the state-of-the-art seed storage involves a container like a cloth bag that will breathe. And these will be kept in a seed storage vault just above freezing and at very low humidity. That's beyond the means of most of us. If you're not planning to store seeds for very long, I think once they're thoroughly dry, you could just store them in things like paper envelopes or paper bags in a, you know, inside your house. I wouldn't use an outdoor un-air conditioned building that gets really hot, because some seeds would lose viability very rapidly. Let's see. Please ignore this. Any tips on improved or unproven instructional resources for keeping alive healthy native milkweeds in containers? They start but always seem to fade or die off. Let me see if the rest of my talk won't address that. I use gallon pots, Let's see, Leslie, thank you. How long can they stay? Okay, that's a discussion between folks, so we'll just let that stand. Here's another method that was used. This is, as far as I know, the first attempted restoration of a native grassland in Texas. It was done by Bob and Mickey Burleson. Bob was the director of TPWD. And back in the 1970s, and there were no commercial sources then of any of these native grasses or wildflowers. And they had some cotton fields that they wanted to restore prairie on in Bell County. Well, they had access to some fragments, tiny fragments of extant prairie. And since they love native plants, you know, they would go, you know, looking for wildflowers there. And they got permission to go, and they would just basically cut hay in these fragments and then take it back to their restoration site and spread it out and then just pack it down. And I have to say that that method, and I have to mention that they would do this repeatedly and at different times of the year, so they get a broader spectrum, a broader sampling of the plant diversity. And I've seen plants that they restored there that you cannot buy, like that Pediomelium in the upper right, and the native Echinacea, not the Echinacea purpurea that's only found in Texas. It's only found in around Texarkana. It's essentially not a native Echinacea, but that's the only one you'll see seeds of in most places. Germination techniques. Okay, another use of appropriate technology. A lot of wild plants have dormancy mechanisms that really work for wild plants, because they don't allow all the seeds to germinate the first time it rains, or the second time, or the third. That seed germination from one good crop of seeds that might only happen once every 10 years, it's going to be spread out over 10 years. So a few germinate every time it rains, because even the plants don't know if this is really going to be a good year to germinate, or are we about to go into a long drought, or is some animal going to come in and eat up all your seedlings. but seed dormancy makes it difficult for us when we want to grow them. One of the very useful methods I've found, which was taught to me by Hoverson. Dr. Hoverson was a plant pathologist in the Rio Grande Valley and one of the first people to grow native plants down there. And we call it aeration. We put the seeds in a mason jar of water with a fish tank aerator, and you just aerate the seeds and change the water maybe 3 times a day. Screen them out, put them back in. And most species, after 1 day of germination or aeration, they can be ready to germinate. Some, and I've grown the Manfredas this way. Notice that you guys planted Manfredas with Don from freshly collected seed. Now, I'm not 100% positive, but I believe that possibly by now, a lot of seeds, once they're a month or 2 old, they'll go into dormancy, and that dormancy may last a long time. So another species of Manfreda that I had grown, Manfreda longiflora, which is a very rare plant, we would want to use seeds that were collected in the previous year. And then I would aerate them and then plant them very shallowly in a sandy soil mix. Just keep it moist and humid, and those seeds came up great. You've all heard of scarification. Many plants have hard seed coats. And, you know, if you're working on small quantities, you can use something like 100 grit sandpaper. That works pretty well if the seeds aren't too small. Now, on large scale batches, when we had to scarify millions of seeds at a time, no, we were not going to individually scarify those with sandpaper. We used technical grade sulfuric acid, which is 93%. And you have to do tests to make sure that you know, there's going to be a Goldilocks level of scarification with acid. You know, not long enough in the acid, and they won't be scarified. Too long, and they're dead. So you got to work that out in advance. And working with sulfuric acid has a lot of safety precautions. It's not for the faint of heart. I will say, some people said, "Oh, can we use battery acid?" And the answer is no. I've tried that. It just doesn't scarify the seeds. It's too diluted. Stratification. You'll hear a lot about cold stratification. I hear this frequently with milkweeds. Why? Because the people who first got interested in milkweeds were in the Midwest, and they wanted to grow milkweeds for monarchs. It's ironic, right? Because that's precisely where people have been learning how to kill milkweeds for decades. But anyway, so we want to grow milkweeds. Well, milkweed species and ecotypes in the Midwest need a cold, moist stratification. The seeds are held in a moist medium like vermiculite in refrigerators for a period of months, and then they'll germinate. I have tried this with milkweeds that I collected in the Austin area, and they mostly rotted before I could plant them. Whereas when I simply stored the seeds for a period of months, they came up fine. Now, we had a project with the South Texas Natives group, and they were looking for milkweeds that could be produced commercially for habitat restoration. And they worked with the Zizotes milkweed, which is really the best monarch host plant in South Texas. And they reached the same conclusion. And I know Don himself created his own Zizotes seed production plot, and he found the same thing. The seeds will germinate once they're, I'm thinking, a couple of months old. They'll germinate without any other treatment. You don't need to cold stratify them. So I would think of that as a warm stratification. Some of the species that I worked with, such as the endangered Walker's manioc, that thing needed about a year of warm stratification before it would germinate. The opposite of a stratification to overcome dormancy are species that have what we call recalcitrant germination, which it doesn't seem like the right word to me, but what it means is these are species that lose viability very rapidly. One of the plants that we grew down in the Rio Grande Valley was the chapotillo, Amyris texana. And we could collect the seeds and depulp them and plant them the next day, and they would germinate really well. And after a week, they were losing viability, and within a month, they wouldn't come up at all. And we tried everything, but they really appeared to be dead. Sometimes dormant seeds can be stimulated to germinate with gibberellic acid, which is a naturally occurring plant hormone. You just have to be careful, and again, you have to work out the right concentration, because if you use too much gibberellic acid, the seeds will germinate and then they will grow extremely long and then just fall over and die. So once again, the aerator method that I described is a great way to leach inhibitors out of seed coats. It works for a lot of species. Many plants of Texas, and particularly a very large representation of our rarest, or rather our plants that are becoming or have become rare, are fire-following species. These are plants of grasslands and savannas. They have an adaptation to fire. You know, if you germinate right after there's been a fire, that's a great time to come up, because all the grass competition is gone. So there's lots of native plants that have this very specific requirement, and they will germinate after a fire. Well, it's been known for many years that such species can be exposed to smoke water, and you can get them to germinate. And it wasn't discovered until some Australian biochemists in 2004 actually isolated these substances called kerrikins that are produced whenever cellulose burns. You could buy kerrikin for something like $10,000 a gram, or you can make your own smoke water. And you can see, I made a smoke water generator out of an old barbecue kettle there, and the chimney was filled with a coil of hardware cloth, and I filled it full of grass clippings and set them on fire and covered it. And as the smoke came up the chimney, I poured water down through it, and it was collected in the mason jar that you see at the left or the right there, and it worked very well. Of course, Monica Mackley, if you know her, she works for San Antonio Express or something like. She's a journalist, and I told her about this, and she announced to a large group of people that I was advocating that if people want to conserve monarchs, they needed to smoke grass. Some species germinate best at or very close to the surface, and an example is Cenizo. And you often hear people say, "Oh, Cenizo, you can't grow it from seed. It doesn't work." And part of the problem is the seeds are very, very small, and they're produced in some capsules that are only about maybe not a quarter of an inch tall. And it's another case where if the capsule is green, the seeds are not ready. If the capsule is ready today, tomorrow it will open and there won't be any seeds left. So people will go collecting these little capsules and they don't realize there's no more seeds. So you really need to watch the plants and possibly visit every day and get the capsules just at the right moment. And after you process them, you'll see these tiny little seeds. They're about a millimeter long, if that. And they germinate great. What I'd like to do, of course, I'll give them that aeration for about a day, and then mix them with some sand and just spread this over the surface of a sandy soil mix that I have in a Rubbermaid tub. And I'll drill some holes in the bottom of it for drainage. So you've got a couple of inches of soil, and you have the seeds on the top. And then I just get a glass jar and just roll it over the top to just pack the seeds into the surface. Make sure it's moist but not wet, and then I just cover it. And the seeds will start coming up, and they're very, very tiny. And they'll come up by the thousands, and you kind of just have to, you're going to have to be real patient and real delicate with them. But after several weeks, they'll get up to about this high, and they'll have, several sets of true leaves. And at that point, they can be transplanted to containers. What should we grow native plants in? Well, for some reason in North America, everybody just wants to buy very expensive bags of commercial potting soil. In my experiences working in the Rio Grande Valley, native plants that were grown in potting soil often didn't grow very well in the first place. And in the second place, even if they grew well, when we planted them outside in a wild, natural soil, it just wouldn't establish. They just died. And my reasoning is that in part, there may be huge differences in the acidity, the pH of commercial soils, which have a lot of peat moss in them. They're usually like down around pH 6. The natural soils were pH 8, maybe 100 times more basic. But also, the commercial soils may either be lacking mycorrhizae, or they may have the wrong mycorrhizae. Mycorrhizae are soil fungi that most plants have to associate with in order to establish in the wild, in the real world. Maybe not in a gallon pot where you're adding fertilizer and everything, but in the real world where the plants are competing with other plants, they've got to have mycorrhizae. The photographs you see on the right I actually took. These were part of my master's thesis. I was studying mycorrhizal fungi that were important for restoring coal strip mine spoil in the Midwest. And also many of our legumes are nitrogen fixing species, but they require the right type of rhizobium bacterium that occurs in the soil. Well, if you can make a soil mix based on the type of soil where the plants live now and where you hope to plant them in the future, you basically you're buying all your inoculum for free, or you're getting your inoculum for free. And you may not have to use a very large sample of soil extracted from a wild population. Maybe 1 gallon of wild soil with maybe mixed into 100 gallons of natural soil. And typically we would mix that about 50-50 with vermiculite. And most, if not all, native plants grew really well in that. Air pruning and the type of container that we use is really important. You can see on the left, there's 3 seedlings grown in plant band containers. These are biodegradable carton containers, and they're open at the bottom. Behind them, you see the notorious gallon pots, which have a flat bottom. And you can see the photograph on the right, you see a kidney wood seedling that was grown in a gallon pot. And you can see that the root, when it came to the bottom, started spiraling around on the bottom. Plants with deformed root systems frequently do not do well once they're planted in the wild. You can see the seedling in the root system of the seedling that was grown in the air pruned container the initial taproot, when it hit the bottom, went dormant, and that released a lot of lateral roots. A root system like that, when it hits the ground, is ready to grow and will produce a healthy surviving plant that grows very fast. On the left, you see an Anaqua seedling that was grown in a container that was 8 inches tall by 1.5 inches wide. And on the right, you see a 1-year-old Anaqua grown in that type of container, 1 year later. Now, that was never irrigated. We just stuck it in the ground at the right time of the year and walked away. And after a year, it was taller than a person. So these methods work. We also have to consider the cost benefit of the type of container we use. Those 1.5 inch plant bands that I mentioned, you can see a trailer there that's 7 by 17 feet, and that would hold 7,776 of those seedlings. If we were to put, if we grew our plants in those 1-gallon pots and transported them in that same trailer, we would only be able to transport 685 seedlings. So you're moving 10 times as many plants per trip with that smaller container. And the end result, even if the gallon pot was as good as the plant band, it's economically it's failing because of all the extremely elevated cost of just shipping your plant to the site. You also have to consider how many seedlings a laborer can plant in a day, and also your nursery space. Here's another example of an air pruning container and a setup. Here you see Texas ebony seedlings in 1.25 inch wide containers, and you can see the hardwood cloth that they're suspended on. So again, the root system goes to the bottom, goes dormant, then you get lateral roots, and then they go dormant, you get more lateral roots. If we accidentally left those seedlings on the ground over the weekend, they were already growing roots that were entering the soil. So seedlings grown in this manner have root systems that very quickly after they're planted will start entering the soil and will establish. And it's the only way you're going to get seedlings to survive in a dry land situation where you finally get that good rain and you plant, and that may be the last rain they get for a long time, as Don can tell you. That's the end of my prepared talk. It was way more than you bargained for. Several people have already passed away, apparently, or fallen over. Here's my quiz plant, and then we'll get to some chat questions. The quiz, for anybody who can identify the plant in this photograph, I will give you some seeds and you can grow them and plant them on your property. Now let me look at the chat. Let's see. Let's see, aeration the meeting. Okay, good. Don says the meeting will be posted on Native Prairie's YouTube channel. Does the aeration only method work with seeds that need stratification? No, I used aeration for almost everything we grew. There's one exemption. There are some seeds, for example, if you've ever grown ruellias or if you've ever grown basil, these are two plants that the seed coat has this these substances, these carbohydrates, that as soon as they get wet, they become mucilaginous and they increase in volume like 100 times. And so if you try to aerate ruellia or basil for that matter, you get what looks like a mass of frog eggs that are all stuck together and you will not be able to work with them. Other than those exceptions, We used aeration for just about everything. And you just have to be aware there's some species that will imbibe very rapidly. Texas kidneywood is an example. Or aerate them overnight and plant them the next morning.
Chris, I see Michael Davidson has his video on, so he may have a question. Michael?
Oh, sorry, that was a mistake.
Ah,
Well, this would be a good time for anybody with questions to turn on your audio and video and ask them of Chris. So are there any more questions out there?
let's see, I'm reading a few more chats. Somebody asked how long before Manfredas go into dormant. I think they're referring to seed going into dormancy, and I'm not sure. And I'm actually not sure if they do go into dormancy. That was my impression when growing Manfreda longiflora, but I'm not positive about that.
Well, that question was for me, and I was said about that Dawn seeds had germinated pretty well because they were kind of fresh. But we're going to be doing that project in September, so it'll be another month or so before we plant them. So I was wondering if we need to do that aeration or any of that.
Well, I think whether or not they are dormant, their aeration is just a great way to get the seeds to absorb water, and the air prevents them from suffocating. That's part of the reason for doing that, is that once seeds imbibe water, they start respiring very rapidly. And if you have a lot of seeds in a mason jar and they're all germinating, they will quickly exhaust the oxygen, and then they actually suffocate just like we would. So by using that fish tank aerator, you make sure that once they do germinate, they're alive. And let's see.
Chris, I saw very on a question that asked how far north is the range of the Manfreda giant skipper?
It parallels the range of the 4 South Texas Manfreda species. So about up to San Antonio and kind of off towards the coast and then down to the Rio Grande Valley. And we know that both the Manfredas and the Manfreda giant skipper are in northeastern Mexico also. There's a question, where in our prairie should we plant our Manfredas? Sandy, dry, wet, rocky? No, Manfredas, they want as much sun as they can get. The Manfreda tuberosa, the Texas tuberosa, maculosa, most often is found in caliche, just in shallow soils over caliche. But some populations are in deep, loose sand, and it is also known from rocky soils. So look for dry, sandy, or rocky, and as much sun as possible. Ipomoea cordato triloba, you have the right genus. Snake herb, Ruellia, prickly pear. Any hints to germinate heartleaf hibiscus? Yeah, that's a tough one. And many species of Malvaceae have very strong dormancy mechanisms and require multiple methods. I think that scarification may be helpful. And some people who had success growing it would put the seeds in a moist medium and like in a Ziploc bag and leave it where the sun would shine on it and it would get really hot. And I don't know how long they actually did that, whether it was just one day or several days, but they told me that they could get the seeds to germinate after that. But that's one, I would say you definitely want to use seeds that are at least a year old. If any of you know Benito Trevino, he grew a lot of the plants that we planted in our wildlife refuge, and he may share some of his techniques with you. Veronia Bowlin, any recommendations on harvesting seed? No, but I have in my landscape Vernonia Lindheimeri, and it's just fairly typical Asteraceae. When you collect the seed heads, make sure they're fully mature. You know, peel open samples of them to see if the seed is still in there, it hasn't blown away, not still green. And again, you can use a variety of screening techniques and air screening to, you know, get a more pure sample of seeds, but you're always going to have a lot of chaff with it. And it should germinate pretty readily, planted at or very shallowly, I mean, at the surface, or maybe, you know, like a very fine covering of sand. Just keep it moist. A lot of Asteraceae that I've worked with don't seem to maintain viability very long. Although I have the Vernonia that I used, I think I germinated seeds that were perhaps a year old. So you can try that. With warm stratification of Zizotes, is that done at room temperature? I think you could even store the seeds, you know, outdoors, but just keep them dry and well aerated. You know, like, yeah, you don't want to have them in a closed container. Don, how did you tell us what method you used to germinate your Zizotes?
Well, we had the best results when we collected some Zizotes along the highway and we planted them within 2 weeks. We got easily a 90% germination rate. So we didn't do any kind of stratification. We did nothing to them except plant them about 2 weeks after we collected them. Others I've kept for a long time and planted them, and we still get a high germination rate.
Mm-hmm.
So I'm not sure you need to do anything other than put them in soil and keep them moist.
Yeah, I'm not surprised. And I think that's what the South Texas natives people found also, that they definitely did not need a cold stratification. Now, it's interesting that there are some milkweed species that occur in central Texas and extend their ranges further north. And I think you may find that those more northern populations do need cold, moist stratification, and the central Texas ones don't. And that's another very good example of ecotypic variation. And that's why we want to plant native ecotypes, not just native species. Now we have a winner. The plant I showed you is Lindheimer Morning Glory. So Jerry Morrissey, if you would like some seed, I collected, I think, 11 seeds last fall. It was one of my personal holy grails. I had never seen this plant, and it's a fairly rare endemic of central Texas. And I searched until I found some, and I collected a few seeds, and I have 6 plants right now. And they've been flowering and they're producing seeds. So, and actually, if any of you would like to grow it, email me, and within a few months, I should have a pretty good collection, and I'll be happy to send you some seeds. And by the way, the technique on that is you do need to immediately, prior to germinating them, sandpaper the seeds very lightly. Use like 100 grit sandpaper. You can hold the seeds in a forceps and just lightly scarify it until you can see the light inner color of the seed. And then I aerated those overnight and I aerated them for perhaps up to 1 day, and you know when they're imbibing because they will swell up. And at that point, you want to plant them immediately.
Chris, I might say we have really kept people longer than normal. This content was, I think, outstanding. And congratulations to Jerry for winning the prize. Anybody that stays on any longer is on their own time. But are there any other questions?
Oh, somebody says, Kath, it's Julia, right?
Yes.
Yes. At the original homestead of Lindheimer, New Braunfels, Lindheimer Morning Glory was growing in the garden. My mom lived down the street from the house and saw all sorts of interesting and probably rare species. Wow, that's a wonderful story. I'll have to go back there and you can visit the Lindheimer house, right?
Yes, you can. It's a really small house on the river. It was really close to the old was it a grist mill, and one of the original grist mills in New Braunfels, and it's real close to the downtown. And yeah, they have a museum. You have to probably set up an appointment to go do the tour, but they have a lot of the original furniture in there, and then they've collected stuff from when he was alive, things that would have been in the home. But yeah,
There's
it's
a homestead down the street that they've not changed anything in the yard. It's the next block over. You know which house I'm talking about?
no.
So on the next street over, it's the second house on the right. It's the original homestead from like 1890. I bet it didn't change the fence in there.
Yeah, but no, New Braunfels there's still some homes, that probably have the original stuff in the yard or wild native plants in there that they haven't done anything to them yet. Nobody's bought the house yet. But anyways.
Interesting.
Okay, any more questions or comments?
No.
If not, let's thank Chris for a fantastic presentation.
Yes,
I
thank
think
you.
now we know why U.S. Fish and Wildlife Services is giving him that award this year.
Mhm.
Oh, I didn't show it to you. It's really cool. It's a piece of handmade glass. Isn't that cool? I told you, Don, it's rough on the edges like I am.
Mhm. Right.
It's nice. Well,
Okay.
thanks for you guys for inviting me and for hanging on so long. And again, if anybody can use the presentation, we can provide that to you. And any additional questions you have, again, just email me. Let me go back down to my contact.
And anybody wants to put in practice what we learned tonight, come out to the Kirchoff Prairie. I think that's on September the 11th. So see
Yep.
you there.
Alrighty.
Okay,
Bye-bye.
thank you all.